Really quiet. Don't forget, quiz. Do it Monday morning. And then after that, there'll be another quiz that we do again on Monday morning. That's the Monday of Thanksgiving week, which is the last lecture before exam 3, last lecture day before exam 3, which hopefully is not a lecture day, but a question day for you. Because I know everybody's going to study every day of Thanksgiving break. Prepare for exam 3 and the final. That's what you should do. Don't watch the wall. Study. A little bit every day. Helps a lot. Okay, so what we're going to spend most of our time and the rest of the semester on is talking about the cell cycle and the control of the cell cycle. So the regulation of the cell cycle. Why and when your cells actually go through the process of cell division. And there's several proteins that are involved in this process. It's a cascade of events that number 1 does something, so number 2 does something, so number 3 does something, so number 4 does something, number 5 does something, and number 6 does something, and then something happens. They're not called number 1, 2, 3, 4, 5, 6, 7. They have other names, so it'll be more confusing. Hopefully will be okay. But the cell cycle is an important part of most of the cells' life in the body because they undergo the process of mitosis. Remember, meiosis makes gametes, and gametes is an end in itself. Those cells don't undergo any more cell divisions. But for mitosis, we need to make copies of ourselves so those cells can go on to make more copies of themselves and continue the existence of that cell line. So again, we start with a diploid cell and we copy our DNA and separate that DNA to make 2 more diploid cells. And for us, one of the most important things we'll talk about is interphase, which is when cells are not actually dividing. M phase is when cells are dividing, and that's a very short time period of the cell cycle. Most of the time is done or spent in what we call the interphase activities. And one of the most important things that happens during interphase is the copying of DNA, this process that we call replication. And a lot of what goes on during the cell cycle is designed to make sure that this important step happens correctly. Copying DNA and making sure the DNA gets copied correctly is a really important aspect of regulating the cell cycle. And as we'll see, if there's a problem, hopefully we can stop things. I thought there was a slide in the middle between this slide and the last slide. Apparently it's a slide from earlier. So we'll just write it on the board. Because it's not a bad thing to have this picture over here to look at for the entire class period. Cell cycle is a continuous process. Once a cell completes the cell cycle, it re-enters the cell cycle. And where things are not as important as where they are relative to each other. Which is to say, it doesn't really matter where you start, it just matters that you go in the right order. But typically we say that we start in G1 phase and we progress to S phase, then G2, then M phase. Just to sort of help us think about the transition from one phase to the other, we'll draw all these little arrows to say that this is the way the cycle goes. S phase is when DNA is replicated. That is always a very important thing to remember. G1 and G2 are called gap phases. And as a gap phase, there are 2 general ideas about what goes on during these periods. One is the idea of recovery, just sort of getting over the last thing we did. Like mitosis is a fairly energy intensive process, fairly sort of rigorous process. And we gotta need to rest, relax, chill out for a second, before we get ready for the next big thing that goes on, which is replication. And so you're sort of getting over what you just did, and you're getting ready for the next thing that you're going to do. That's what we want to think about with G1 and G2 phases. Preparation for the next thing and recovering from the previous thing. One of the things that's not in the slides yet, but will be important, is there is a fifth phase of the cell cycle, what we call a G0. The cells can leave the normal cell cycle, enter G0, but they can also re-enter the normal cell cycle. Its position here at the end of G1 is not important. It could be anywhere, it's just this is where it sort of fits, based on my picture. G0 is considered a non-dividing state. Which for us is kind of like a waiting. You know, waiting. And maybe there's something wrong. Maybe there's not anything wrong. We are waiting. Paused is another sort of good idea to think about the G0 state. Is the cell is paused in the cell cycle. Some cells in our body are permanently paused in G0 because they never divide. Some cells never go to G0 because they're always dividing. We just need to consider this. But also G0 is a state that we can use for repair. So while we're paused, typically that means repair can be done. That's one of the things that we'll think about as we go forward is there are places where we can stop the cell cycle for anything bad has happened, trying to fix the problems. If they're fixed or fixable, do it and continue on. But sometimes we just need to scrap the whole thing and start over. DNA replication is what we call energy intensive. Which means it takes a lot of energy to do. You have to make all the proteins needed to do replication. You have to spend energy forming all those phosphoester bonds. Energy intensive process. Mitosis and cytokinesis are also very energy intensive processes. It takes a lot of energy to move the DNA around and to do all the other things that need to be done. The reason why I bring this up is G0 sort of makes sense to happen early on. If you're going to pause before you spend any energy. Because once you sort of started, you're committed to sort of going through the whole thing until you get back to another sort of pause. Again, M phase is both mitosis and cytokinesis. So the M doesn't exactly stand for mitosis exactly. But it kind of does. We can't forget that cytokinesis is not mitosis and vice versa. So mitosis, like meiosis, is just the division of the DNA or the division of the nuclear contents, and cytokinesis is the thing that's going to physically separate the cytoplasmic contents of the cell and actually give us 2 new cells at the end of this period. In M phase is a very short process. Interphase, the G1, S phase, and G2 are a very long part of the process. In this diagram here, they're sort of breaking it down into a 24-hour clock. You can see 1 hour of 24 spent actually doing cell division. The other 23 are getting ready to do cell division. So again, we spend a lot of time prepping. Yes?
Should we remember how long each individual section takes, or just the order it happens in?
Just the order. The numbers for time is put it in just perspective of like how short or long things are. But all you might need to know is interphase is the longest part of the cell cycle by a wide margin, and M phase is by far the shortest part of the cell cycle. So just think about interphase, G1, S phase, and G2 are where the cell spends the most time. How much time of that is not important. It's not 90%, 80%, just the most time is spent in interphase. For meiosis, we talked about the steps of meiosis. prophase, metaphase, anaphase, and telophase. And again, we use the same language with mitosis. Start with the prophase, move on to metaphase, continue to anaphase, and telophase, and then eventually to cytokinesis. And what we remember, hopefully, from meiosis will help us with mitosis, with three really important exceptions. Meiosis is about creating genetically diverse gametes. So crossing over and independent alignment are really important in the process of meiosis and creating genetic diversity. With mitosis, there's no diversity. We are trying to make an exact duplicate. So no crossing over, so no synapsis, no independent alignment, no tetrad formation. None of that stuff happens in mitosis. But basically everything else does. So we'll remind you about what happens during these phases. Again, no crossing over, no genetic diversity. Let's make identical copies of ourselves. Again, hopefully you remember some things about prophase, metaphase, and anaphase, and telophase from before. During prophase, again, we see our chromosomes get smaller, so they're more easily moved around. We have to get rid of the nucleus, because that would restrict the movement of the DNA inside the cell. We also need to make our spindle fibers. And those kinetochores need to be attached to the chromosomes, which means we kick out one of those histone proteins, replace it with the central protein, make the kinetochore connect, and add those microtubules to the kinetochore so we can move things around. If you forgot about histone proteins and central, that's fine. It's not an important part of exam 3. But the position of this material here at the end is to remind you of that stuff, because it will come up on the finals as kind of like a review of meiosis, even though we're talking about something else. As we think about prophase, and one of the things that I want you to think about with meiosis is prophase and telophase are opposites of each other. What we do during prophase, we undo during telophase, because we don't need the DNA to be in the same state anymore. Making the DNA smaller makes it easier to move. Moving the nucleus makes the DNA easier to move. Adding the kinetochores and the microtubules, what we call the spindle fibers, allows us to move the DNA. Everything is done so that we can move the DNA around during metaphase and anaphase. When we were talking about meiosis and prophase, I started to give you this example, but stopped because it wasn't in the slide. So here it is. This is a sort of an analogy of the amount of condensation that occurs during prophase. So they use the analogy that a chromosome is the diameter of a spaghetti noodle. And if the chromosome is as long as chromosome 1, that would be a spaghetti noodle that's 25 miles long. Can you imagine the spaghetti noodle being 25 miles long? A cell is basically like a micron across, a micrometer. What's annoying about this is it's metric and standard together. Millimeters, miles. I mean, come on, dude. Pick a thing and use it. Anyways, it's pretty thin and very long, which is what we want to think about with chromosomes. But as we go through the process of for prophase specifically, that 1 millimeter thick thing becomes 2 feet wide. Again, switching from metric to standard. Who can do that in their head? Anybody? And what would be really funny is if instead of being 6 feet long, it was 2 meters long. So you go from miles to meters and millimeters to feet. But think about that. Something that's 1 millimeter wide is, that's that little thing. 2 feet wide? 2 feet. These things get really short, but also really thick. Which is why they get visible. That's why we can actually see them. Don't worry about trying to memorize this. It's in gray. These in gray are not worth memorizing. But it's fun facts. Fun facts. Remember, metaphase is all about the DNA moving to the middle. During mitosis, there is no synapsis, so each chromosome, maternal and paternal, are acting independently of each other. So instead of having 23 tetrads go to the middle of the cell, as we do during meiosis, we have 46 chromosomes going to the middle during mitosis. Remember, each one of those chromosomes is a sister chromatid pair. And as we get to the middle, they sort of pause for the briefest amount of time, and then anaphase happens where those chromatids of the sister chromatid pair get pulled apart. So we see the separation of sister chromatids occur during the anaphase of mitosis. If you remember anything about meiosis, maybe you remember that it is during meiosis 2 that the sister chromatids are separated. So we say that mitosis and meiosis 2 are more or less analogous to each other, that they do the same sort of setup. Sister chromatids move to the middle, or sister chromatid pairs move to the middle, and then sister chromatid pairs are separated during anaphase. And DNA goes to the middle, move to the middle so we can separate things all evenly, pull them apart, and then we split the cell in half during cytokinesis. But before cytokinesis, we have a telophase, which again is the opposite of prophase. One of the things that I also talked about during meiosis is metaphase and anaphase are also kind of opposites. Moving to the middle and moving away from the middle is an opposite type of idea as well. We coalesce to the middle so we can separate away from the middle. Get small, move to the middle, move away from the middle, get big. And then cytokinesis happens, which is dividing the rest of the cell. If you want some cartoons of this, you can go to the next couple slides and see some drawings of mitosis that use the same format as previously with meiosis. But what you won't see is you won't see independent alignment, so there's only one metaphase possibility. Sister chromatids should be completely identical to each other, so it doesn't matter how one lines up relative to the other, it's the same on left and the same on right. And for the most part, mitosis is not a big part of exam 3. The cell cycle is the biggest part of what we want to talk about. And as we go through the cell cycle, we will see some important things happen. And they're going to revolve around what we call checkpoints. And one important checkpoint will be right here between G1 and S phase. Something important is going to happen here that we need to sort of think about. There's another important checkpoint that will occur sort of right here at the end of G2 phase. And these are places where we can just make sure everything's working fine before going forward. It's not exactly a checkpoint, but there will be an important event during M phase that we'll look at. And again, it's not a checkpoint exactly, it's just an event. It's like what we call it. We'll call it an event. Important event, we'll just call it an event. We'll see what it is as we go forward, but there is something important. It happens right here, somewhere in the middle of mitosis. And here's where we're going to spend our time for most of the rest of the semester, is talking about what goes on here, and how is this controlled, what goes on here and how is it controlled, and what goes on here and how is this controlled. It's going to introduce some of the different types of regulatory mechanisms that we haven't talked about yet. So we're no longer really talking about transcriptional control or translational control. The vast majority of things that we're going to talk about are going to be post-translational control mechanisms. How do we control a protein that is already around? And if you remember, the very last slide in that regulation set of slides talk about these sort of three things that we see very commonly occur. One is going to be protein phosphorylation or dephosphorylation. There's an important process where we use proteolysis as a way to control protein activity. And the other is what we call protein sequestration, where a protein binds to another protein to prevent it from doing anything. We have ways to control proteins that are already around. Even if we stop transcription and stop translation, some of those proteins are already out there in the cell. We need to be able to control those as well. And this is all wrapped around the idea of cancer, because it turns out that a lot of cancers are caused because of a breakdown in the normal cell cycle. The things that are controlling how a cell and when a cell divides are broken in tumor cells, which cause the disease we call cancer. So one of the more important ones is the protein called P53. It's like the master control protein for this process. Now, I apologize about only putting these slides up like 20 minutes ago. Actually, now it's probably 40 minutes ago, but I was still sort of rearranging things because I didn't like the way they went last semester. It's there now, and hopefully you have access to them. Starting out, we have two definitions. Senescence. You ever heard of the word or the idea of senescence before? It's sometimes hard to define, but we typically like to think about it as a biological clock. Senescence is the idea that everything lives and dies in its time. And when we look at cancer cells, they typically don't have the normal senescence that a cell should have. So they're living longer and doing things for more than what they should be doing. And senescence is every cell has a life that they live, and once that life is over, they should die. And they typically die because of the process we call apoptosis. I'm old school, so that pop is really important because it tells you what's going on. Cells are dying, so think about cells popping. And something pops, that's pretty much dead. When I was a student, we didn't have these touchy-feely bullshit, worrying about people's feelings. So they called apoptosis cell suicide. And now, I guess, suicide has some negative connotations, so they call it programmed cell death. So much better, right? I guess because suicide is a choice, apoptosis is an inevitability. Not really a choice. You're going to die eventually. That's what apoptosis is. You're going to die eventually. But wrapped in this idea of you're going to die naturally sometimes is the idea that we can make you die early if you're bad. So typically things that are bad are damaged cells or infected cells, things that have viral infections or bacterial infections, or tumor cells. They can try to get them to undergo apoptosis so they don't cause any problems. And Star Trek should be invoked here if you know anything about Star Trek. The good of the many outweighs the good of the one. So save the organism, the human, by sacrificing one or two cells here and there is actually a good thing. So every cell sort of lives a certain amount of time. As we think about the final exam, senescence also refers to telomere length. Remember, telomere length controls cell divisions. As telomeres get shorter, the cell gets to the point where it's sort of ending its lifetime. So we're going to revisit telomeres again a little later. There are two important things that control when a cell will divide. One of them is called contact inhibition. The other is called anchorage dependence. They're opposites that sound exactly the same. So a lot of people have problems sort of getting these mixed up. Contact inhibition is basically what it sounds like. Contact, touching another cell, is a signal not to divide. Which means if you're not touching another cell, that's a signal to divide. And one of the most easy examples of illustrating contact inhibition is when you cut yourself and you create a wound. And the idea that you want to think about is what happens when you cut yourself. You have a place where you're missing some skin cells. And so normally all these skin cells that are right there at the wound site should be touching cells in here. Because there's missing, these cells no longer have contact inhibition, so they start to divide. You sort of get this cells dividing, which means you start filling in the gap. Once they start touching each other, they should stop dividing, because that's what contact inhibition says. Don't divide if you've already sort of filled in the missing area. And cells, these eukaryotic cells typically only grow in one dimension, so they don't typically grow up unless they're doing something wrong. Which is a mold. I mean, have a mold or know the mold? It's the little growth that comes up out of your normal skin. That's doing something wrong. That's why moles can sometimes be tumorous, because they're doing something they're not normally supposed to be doing. But contact inhibition is cells that are no longer touching their neighbors are triggered to divide to fill in the missing cells that they have lost. And it's most often associated with skin cells, because that's the thing we cut most often, and it's very easy to repair. Less easy to do with other organs in the body, but for skin, very easy to sort of fill in the gaps. Because once you sort of meet in the middle, you stop dividing again, because contact inhibition reexerts itself. So we don't want those skins to keep dividing and building up, like a skin tag or a mole. So this idea of your skin cells are not growing the right way. Anchorage dependence is a way to keep cells from dividing if they're in the wrong location. And one of the worst types of cancers is what we call metastatic. Is where cancer starts in your lungs and then spreads to other organs in your body. Now normally, if a lung cell leaves the lungs and finds itself in the liver, anchorage dependence should keep it from dividing in the wrong location. So nothing bad should normally happen because of anchorage dependence. Cells that sort of lose their contact to their other cells should be inert, normally speaking. But if they're in the liver, we can make more liver cells, because maybe you need more liver cells, or more heart cells, or more lung cells. But only in the lungs, only in the liver, only in the heart. So keep the cells in their proper location, and only divide when they're there. And that's what anchorage dependence says. Where your anchorage is where your house is, basically. Only divide when you're home. And only divide if there's missing things. So these two things control when cells divide. And these things are typically broken. Or one or the other could be broken in a tumor cell to lead to cancer. So we put a lot of regulatory potential into controlling cell division, because again, contact inhibition is keeping you from dividing if you're touching the right neighbors. Anchorage dependence says only divide when you are touching the right neighbors. So we have to control this very tightly, or things go off the rails really quickly. And one of the most important things that we're trying to do with the cell cycle is make sure we copy our DNA correctly. And before we copy our DNA, we fix any mistakes first. So one thing you really don't want to do is copy bad DNA. That means your daughter cells get bad DNA, so you're just making more trouble. So not only do we copy the DNA correctly, we make sure it's correct before we copy it. Right? Never a good idea to cheat off somebody that's getting things wrong all the time, because you're just going to get them wrong too. Cheat off the smart people. You're not supposed to cheat, right? There is another important function. You can see that we also want to make sure that DNA gets separated correctly. And so the idea of chromosome replication happening correctly is what these first two checkpoints here are going to deal with, because they bookend the replication step. This step here, this event that I mentioned earlier, is basically this thing here about chromosome separation, chromatid separation. So we have an idea of what we're going to talk about. We haven't really talked about it just yet. But these are the things we're trying to do: is make sure that DNA gets copied correctly and that DNA gets separated correctly. There's a lot of things that can go wrong, and we can't talk about all of them. This slide tries to summarize some of the things. It's also nice because it does show us the 3 important events. An event that happens between the G1 phase and the start of S phase. An event that happens at the end of G2 before we start M phase. And then something happens in the middle of M phase, which is actually a metaphase, anaphase sort of problem that we have to deal with. So these are the 3 events. Even though they're not listed or sort of labeled, they're sort of positionally there. But these are the 3 things we're going to talk about. But there are other things that are sort of not these 3 things. So if you want to just look at the slide and get some more information for stuff that doesn't affect us, feel free. We're going to cover everything we need to cover. So don't really worry about trying to memorize this, or don't memorize this slide. It might help afterwards. Definitely don't do it early. Okay, checkpoint in G1 and a checkpoint in G2. Again, these are all about DNA issues. So have we copied our DNA correctly? Is our DNA error-free or mistake-free before we copy it? Has anything happened to our DNA after the copying process? These are the things we're trying to fix. Before we copy our DNA, and after we copy our DNA, is there anything we can do to fix any things that are problematic? So here is a slide that actually mentions this G0. Sorry, I forgot this is a subscript, so don't call it go. It's not the go phase, because that's the opposite of what it actually does. It doesn't go, it's a no-go phase. But if something happens wrong in G1, we can leave G1, enter G0 to wait and fix things before restarting G1 and continuing with the cell cycle. Once we're in G2, there's really not a whole lot you can do. It's either continue or undergo apoptosis, which one thing we'll sort of continue to talk about as we go along. This next slide is going to tell you the exact same thing I saw on the previous 2 slides. There's an important thing that happens at the end of G1, at the beginning of S phase. There's an important thing that happens at the end of G2, at the beginning of M phase. And these are the 2 things that will control whether our DNA is correct. We don't deal with this one, so we can ignore this one for us. It's not going to be important. Again, making sure DNA is copied correctly is an important aspect of controlling the cell cycle. It's not just mutations and mispairing we're looking at, or timing dimers, or whatever it is. We could also be looking for, are there any translocations or duplications or deletions that might have occurred, even though we're not doing crossing over. These things can happen. Let's fix everything that we can fix, so bad things don't happen. What I put on the board is an approximation of what I put in this slide here. It was just a little earlier, but again, here it is, so you have it 2 times. But again, the idea of G1 and G2 is recovery and preparation. And that's an idea that we want to expand on a little bit right now. What are we preparing for? And what do we need to do to make that thing happen? During G1 phase, see, we're preparing for DNA replication. What do we need to make so that we can do DNA replication? It's a general question, not a specific question. What do we need to do DNA replication? What does DNA replication? What are all the little bitty things made of amino acids that perform all the important functions inside cells. We need to make the proteins for DNA replication. Which means during G1, are we making DNA replication genes? No, right? Because we don't need them right here. We don't need them here, we don't need them here. We definitely need the DNA replication genes during here. So when we're doing replication, there's gene expression. DNA polymerase has to be made. DNA gyrase has to be made. Mut L, mut S, mut H have to be made. Everything else, DNA ligase, primates, all the other things that are involved in replication have to be made actually before we start S phase. All of this has to go on basically right here. So when we start S phase, we're actually ready to replicate our DNA. So, like from here to here, there's no gene expression of DNA replication genes, but something has to happen to start that process. And that's what we want to talk about. This is where our checkpoint happens, and there's this transition that cell has to undergo. Stop repressing the DNA replication genes and activate the DNA replication genes so that we can actually do DNA replication. So we start that process here. What do you think we need to do over here? And we need to stop it. So this is what we need to think about with the cell cycle. We only need the genes needed for replication during S phase. We make them here at the beginning, and then we stop making them here at the end. If we don't do this, bad things will happen. What do you think might happen if we don't stop expressing DNA replication genes?
Cancer.
Ultimately, possible, yes, but more simply and more straightforwardly, the DNA might replicate again. So we just go in this cycle of we keep copying our DNA, but nothing ever gets made. And that's how you get sometimes multi-nucleated cells, is they keep copying their DNA without doing the cell division. So we could get sort of stuck in a weird place. And during M phase, we do something very similar. Some things will be turned on. No, it's not turned on exactly, but they're turned on. At the beginning of M phase, some things will be turned off as we leave M phase. The kinetical proteins aren't around except start of prophase. The microtubules that are used to move DNA around are not around except during prophase and through mitosis. The proteins that basically cut the cell in half during cytokinesis are not needed during G1 phase, S phase, and G2 phase. They're only needed during when they're needed. So there's lots of this regulation that happens of turn things on at certain times and turn them off when they're not needed. And this is a general discussion right here, just thinking about as we go from one to the other, we see things get turned on or turned off depending on what we're doing. And most of what we're going to talk about is this turning on, turning off that goes on between G1 and S phase. Revisit this idea as we go forward. Sort of keep talking about it as we go forward. When it comes to affecting the cell cycle, there are two broad categories of genes that have been identified. One group of them are called proto-oncogenes. The other are called tumor suppressor genes. Their names are kind of misleading. Or maybe not kind of, their names are misleading. Because it sounds like their only role is either to make a tumor not happen or to make cancer happen. I don't know if you know what an oncogene is. It's typically what we call a gene that causes cancer. That's why people that study cancer are called oncologists. That's where this sort of term comes from. But they don't cause cancer. They make the cell cycle work properly. These don't stop cancer. they make the cell cycle work properly. So when they're broken, cancer is a possibility. And that's what we want to think about is when we break these things, cancer could be a possibility because we're no longer doing things the right way. The things that are controlling the cell cycle are broken. It's like, well, just the red light is out, so it's never on or off, so you like go through it, and then you're gonna get hit on the side because it's broken. You never know who's coming and going. But for proto-oncogenes, they are genes and proteins that will try to make cell division happen. Which means we want to progress from G1 phase to S phase. So they're trying to continue with the cell cycle as normal. Or they tell us to go from G2 to M phase. These things want us to go through the cell cycle normally. So they're pro-cell division. Keep things running the way they're supposed to be running. And because we're pro-life, we're anti-death. So keep cell division going, no need to sacrifice any cells. So keep cell division going, prevent apoptosis from happening, because things should be good. Things are normal, things are good. Allow cell division to keep going and prevent cell death from happening. And you can see that when proto-oncogenes are mutated, typically it gives them, it's either gain of function mutation or a hypermorphic mutation, which means they are really, really, really pro-life and really, really, really anti-death, which means if proto-oncogenes are mutated, something that would normally cause apoptosis won't. Something that would normally cause cell division to stop won't. Keep doing cell division and keep not dying. Keeps them alive longer and keeps them making lots and lots of cells. We see the opposite from things we call tumor suppressor genes. So normally these have nothing to do with cancer whatsoever. They're trying to make the cell cycle work properly. And so their function is to make sure we don't divide when we're not supposed to. Maybe they're involved in contact inhibition. Maybe they're involved in anchorage dependence. Maybe they're involved in making sure the cell, for in G1, it's not doing something that needed for mitosis or S phase or later on. We're trying to control things so they happen the way they're supposed to happen. And if we're anti-life, then it means we're pro-death. Yay, pro-death! Anybody pro-death? Are you anti-life? Nobody's anti-life? It means you're all pro-life? That's a political joke, sorry. Do you see how these things function? There are some things that want us to do cell division. There are some things that say don't do cell division. And if they break, things go wrong. And so typically with the tumor expressor genes, when they're mutated, the mutations are typically loss of function or hypomorphic. So they stop working. So when it would normally cause cell division to stop, it doesn't stop and it keeps going. When it would normally cause cell apoptosis to occur, it doesn't occur, and that cell then doesn't die. All of these two things, again, these two together work to make sure you divide when you're supposed to divide, and don't when you're not. And when you break those two things, you get the wrong thing happening. There's a lot of literature and studies done on proto-oncogenes and tumor suppressor genes. I shouldn't date myself, but when I was applying for grad school, oncogenes and proto-oncogenes were like the newest discovery. So that's what everyone to do, is study proto-oncogenes. I didn't really know much about proto-oncogenes, so I went to this interview, and I shouldn't name drop this, but where I went to interview. I said, maybe we can find a way to turn proto-oncogenes off. And you can't do that. Why not? That's how you can turn them off. I was right and they're wrong, so fuck them. They should have accepted me. Those losers. It's okay with somewhere else doing better. But speaking of studying things, this is not an actual research article. Sorry, it's a story from CNN. But it was about a research article, and this is actually, I can't remember when this was now. I should have written down the year. think it's like 2020. Anyways, there was a group looking at whales. You been anything about whales? They hold water. That's not a joke. You like puns? You don't like puns? Get out. Pun's fun. Anyways, there's a fun line in here. It says, whales, the genes involved in the control of cancer onset and progression were positively selected. And they have 2.4 times faster turnover rate of tumor suppressing genes. Do we have any idea what faster turnover rate means? What does turnover rate mean in general? Like if an employer has a fast turnover rate, what does that mean? Or people quit a lot, or you're always hiring new people. So it turns out that in some organisms, they can make copies of genes. We talked about this already, gene duplication. If you keep sort of making a new copy that's not mutated, and throw away the bad one, so it stays sort of regular for longer periods of time. And that's sort of what these people are trying to say in this article, is that for some reason whales sort of remake their DNA faster or more often than other things. And that might be one of the factors that makes these animals that are really long-lived not suffer from cancer as much as other animals that live much shorter periods of time. Typically when you think about cancer, the longer you live, the more chance you have of getting cancer. But whales live a really long time and they hardly ever get cancer. So they've got something good going for them. And maybe if we know enough about them, we can genetically modify humans to be whales. Have flippers and tails and blowholes. I already have a blowhole. Toot toot. That's probably not the blowhole you're thinking of, where you. Yeah, well, whenever it costs, we talk about diarrhea quite a bit, so I didn't want to risk anything. Unfortunately for you, I know you're really excited to hear about some more stuff. I have to wait until Monday.